Hello students, today we will learn about genetic engineering of plants and its methodology. This video is the third video of genetic engineering of plants and its methodology. So let's begin. Marker genes will start with marker genes for plant transformation. In general, transformation of plants is a low frequency event. Some methods for selecting the transformed plant materials, that may be cells or tissues, have been devised by using a set of genes referred to as marker genes. These marker genes are introduced into the plant material along with the target gene. The marker genes are of two types. First is selectable marker genes, second is reporter genes. So, first we will discuss about selectable marker genes. The selectable marker genes are usually an integral part of plant transformation system. They are present in the vector along with the target gene. In a majority of cases, the selection is based on the survival of the transformed cells when grown on a medium containing a toxic substance. That may be antibiotic, herbicide, antimetabolite. This is due to the fact that the selectable marker gene confers resistance to toxicity in the transformed cells while the non-transformed cells get killed. A large number of selectable marker genes are available and they are grouped into three categories: antibiotic resistant genes, antimetabolite marker genes, and third is herbicide resistant genes. Now let's know about the antibiotic resistant genes. In many plant transformation systems, antibiotic resistant genes that particularly of E. coli are used are as selectable markers. Despite the plants being eukaryotic in nature, antibiotics can effectively inhibit the protein biosynthesis in the cellular organelles, particularly in chloroplast. Some of the antibiotic resistant selectable markers genes we will now discuss. Now let's know about neomycin phosphotransferase 2 NPT2 gene. The most widely used selectable marker is NPT2 gene encoding the enzyme neomycin phosphotransferase 2 NPT2. This marker gene confers resistance to the antibiotic kanamycin. The transformant and the plants derived from them can be checked by applying kanamycin solution and the resistant progeny can be selected. Hygromycin phosphotransferase HPT gene. The antibiotic hygromycin is more toxic than neomycin and therefore can kill non-transformed plant cells much faster. Hygromycin phosphotransferase gene thus provides resistance to transformed cells. Aminoglycoside adenyl transferase AAD capital A gene. Aminoglycoside 3 adenyl transferase gene confers resistance to transformed plant cells against the antibiotic streptomycin and spectinomycin. Now let's know about the antimetabolite marker genes. First is dihydrofolate reductase. The enzyme dihydrofolate reductase produced by DHFR gene is inhibited by the antimetabolite methotrexate. A mutant DHFR gene in mouse that codes for this enzyme, which has a low affinity to methotrexate, has been identified. This DHFR gene fused with CAMB promoter results in a methotrexate resistant markers which can be used for the selection of transformed plants. Now let's know about the herbicide resistant markers. Genes that confer resistance to herbicide are in use as markers for the selection of transgenic plants. Phosphinothresin acetyltransferase PAT or BAR gene. Bialofos bialofos, phosphinothresin, and glufosinate are commonly used herbicides. A PAT or bar genes code for the phosphinothresin phosphinothrysin acetyl transferase which converts these herbicides into acetylated forms that are non-herbicidal. Thus, PAT or bar genes confer resistance to the transplant plants. Enol pyruvyl cyclimate phosphate synthetase. The herbicide glyphosate inhibits photosynthesis. It blocks the activity of enol pyruvyl cyclimate phosphate, that is in short EPSP synthase, a key enzyme involved in the biosynthesis of phenylalanine, tyrosine, and tryptophan. Mutant strains of Agrobacterium and Petunia hybrida that are resistant to glyphosate have been identified. The genes EPSPS or AROA confer resistance to transgenic plants which can be selected. Bromoxynil nitrilase. Let's know about bromoxynil nitrilase. The herbicide bromoxynil inhibits photosystem 2 bromoxynil nitrilase enzyme coded by the gene BXN inactivates this herbicide. The gene BXN can be successfully used as a selectable marker for the selection of transplant plants. Now let's know about the production of marker-free transgenic plants. There is a growing concern among the public regarding the use of antibiotic or herbicide-resistant genes as selectable markers of plant transformation. The products of some markers genes may be toxic or allergic. Antibiotic resistant might be transferred to pathogenic microorganisms in the soil. There is a possibility of creation of superweeds that are resistant to normally used herbicides. A transgenic plant with selectable marker genes cannot be transformed again by using the same selectable markers. A light of apprehensions till now we studied the public is concerned about the safety of transgenic technology, particularly related to the selectable marker genes, antibiotic or herbicide resistant genes. These are these are fears about the there are fears about the safety of consumption of foodstuffs derived from genetically engineered plants. This is despite the fact that so far none of the marker genes have been shown to adversely affect human, animal, or environmental safety. Now let's know about the clean gene technology. What do you mean by clean gene technology? The process of developing transgenic plants without the presence of selectable marker genes or by use of more acceptable marker genes is regarded as clean gene technology, and this will result in the production of many marker free transgenic plants that will be readily acceptable by the public. Some of the approaches for clean gene technology are now we will discuss. First is selectable avoiding selectable marker genes. Theoretically, it is possible to totally avoid marker genes and introduce into the transgene of interest. The transformed plants can then be screened by an advanced technique like polymerase chain reaction and the desirable plant selected. This approach is not practicable due to cofactor. Now let's know about the co-transformation with two DNAs. The transgenic plants can be produced by employing two separate DNAs, one carrying the desired target gene and the other the marker gene. The transformed plants contain both the genes but at different sites on the chromosomal DNA. Traditional breeding techniques can be used to breed grid of transgenic plants with selectable markers. Now let's know about the removal of selectable markers. It is possible to selectively remove the selectable marker genes from the plant genome. For this purpose, site-specific recombinase systems are utilized. Several recombinase systems are in fact available which can be used to selectively excise the marker genes from the plant genome. Now let's know about the cloning of selectable markers between transposable elements. A selectable marker gene can be cloned between plant transposable elements and then inserted. The selectable marker is flanked by the sequence that increase the intrachromosomal recombination. This results in the excision of the marker genes. Now let's know about the reporter genes. A reporter gene may be regarded as the test gene whose expression can be quantified. The plant transformation can be assessed by the expression of reporter genes. In general, an assay for the reporter gene is carried out by estimating the quantity of the protein it produces or the final products formed. Now let's know about the opin synthase. That means OCS or NOS genes. The common opins present in tDNA of type Ti or Ri plasmid, Ti plasmid or Ri plasmid of Agrobacterium are octopine and nopalin, respectively produced by the synthase genes OCS and NOS. The transformed status of the plant cells can be easily detected by the presence of these opins. Opins can be separated by electrophoresis and identified. Alternately, the enzyme activities responsible for the production of opin can also be assayed. Now let's know about beta glucuronidase. Beta-glucuronidase producing gene is the most commonly used reporter gene in assaying plant transformation for some regions. Let's know about the regions. Beta-glucuronidase assays are very sensitive. Quantitative estimation of the enzyme can be done by fluorometric method using substrate 4-methyl amblyophenylpyrrole beta D glucuronide, which is hydrolyzed to 4-methyl lumbeniferone. Quantitative data on the enzyme can be obtained by histochemical means. Enzyme localization can be detected by chromogenic substance such as substrate X-gluc. No need to extract and identify DNA. Green fluorescent protein GFP gene. Green fluorescent protein coded by GFP gene is being widely used in recent years. In fact, in many instances, GFP has replaced GUS since assays of GFP are easier and non-destructive. The screening of even the primary transplants can be done by GFP, which is not possible with other reporter genes. Genes for GFP has been isolated from jellyfish Equoria vectora, which is a luminescent organism. The original GFP gene has been significantly modified to make it more useful as a reporter gene. GFP emits fluorescence, which can be detected under a fluorescent microscope. Now let's know about the bacterial luciferase or Lux A, Lux B genes. The bacterial luciferase gene have originated from Vibrio harvey. They can be detected in some plant transformation vectors. The detection assay of the enzyme is based on the principle of bioluminescence. Bacterial luciferase catalyzes the oxidation of long-chain fatty aldehydes that results in the emission of light which can be measured. Now let's know about firefly luciferase LUC gene. The enzyme firefly luciferase encoded by the gene LUC catalyzes the oxidation of D-luciferin, ATP dependent, which results in the emission of light that can be detected by sensitive luminometers. The firefly luciferase gene, however, is not widely used as a marker gene since the assay of the enzyme is rather cumbersome. Now let's know about chloramphenicol acetyltransferase CAT gene. The CAT gene producing chloramphenicol acetyltransferase is a widely used reporter gene in mammalian cells. Due to the availability of GUC and GFP reporter system for plant transformers, CAT is not commonly used. However, some workers continue to use CAT by a sensitive radioactive assay for the detection of the reporter gene CAT. Now let's know about the promoters and terminators. What do you mean by promoters and terminators? The ultimate objective of gene transfer is that it's a correct expression to provide the desired character or a trait. The appropriate expression of genes is made possible by the presence of promoters and terminators. The DNA sequence upstream, the coding region is the promoter while the terminator is the sequence at the 3' terminus. The promoters are responsible for the commencement of transcription, whereas the terminators ensure the cessation of transcription at the correct position. Promoters possess certain inherent characters such as promoter strength, tissue specificity, and developmental regulations, which determine the efficiency of promoter function in gene expression. Now let's know about agrobacterium derived promoters and terminators. The genes coding for nap, napalin synthase in Ti plasmid of agrobacterium are frequently used as promoters and terminators in plant transformation vectors. Now let's know about the 30S promoter of CAMB. The cauliflower mosaic virus CAMB, in short CAMB, 30S promoter is the most widely used as a promoter in plants. This promoter, a 30S RNA gene, is expressed in almost all the tissues of the transgenic plants. The derived strength of 30S promoter is much higher in dicot compared to monocot. The 30S promoter is preferred for the appropriate expression of selectable marker genes and reporter genes. In recent years, the efficiency of 30S promoter has been further increased by suitable modifications in the enhancer regions. Now let's know about the promoters for monocots. Since 30S promoter is not very efficient for monocots, alternates are used. Ubiquitin 1 promoter or the rice actin promoter are frequently used for high expression of transgenes in monocots. Now let's know about the inducible promoters. In recent years, certain inducible promoter systems for transgene expression have been developed. These promoters differ from the 30S promoter, which is consecutive in nature. There are mainly three inducible expression systems. First is non-plant derived systems. These inducible promoters are dependent of the normal plant processes. A specific exogenous chemical will induce their expression. Example tetracycline, ethanol, steroid, copper. Non-plant derived inducible promoters are useful but not economically viable. Now let's know about plant derived systems based on response to environmental signals. These systems are not independent since they are plant derived and actually form a part of normal plant processes. Plant derived systems respond to a variety Plant-derived systems respond to a variety of environmental signals, that is wound or heat shock. Now, let's know about plant-derived systems based on the developmental control of gene expression. Certain genes that express at a particular stage of plant development have been identified. The promoters of some of these developmental genes are useful for transgene expression. Senescence specific gene expression, abscisic acid inducible gene expression, auxin inducible gene expression. Although plant derived systems are not independent as non plant derived one, they are still useful in plant biotechnology since no exogenous inducer is required. Now let's know about tissue specific promoters. Efforts are on in recent years to isolate promoters that can drive gene expression in a tissue specific manner. The advantage with tissue specific promoters is that the expression is confined to selected tissues that are not consumed by humans or animals. In fact, some tissue specific promoters have been isolated and are in use. Let's know about the transgene stability expression and gene silencing. The genetically transformed plant cells are grown in vitro to finally regenerate two plants. At the initial stages of plant cell growth, some evidence for the success of plant transformation can be obtained, example resistance to antibiotics, herbicides, etc. The integration of transgenes with the host plant genome can be confirmed by some molecular techniques like southern hybridization, polymerase chain reaction, etc. This is usually carried out by analyzing the seeds at T1 generation. What is actually required for the success of plant transformation is the efficient and stable expression of transgene and not just its presence in plant genome. It is just useless to have a desired gene without appropriate expression. Now let's know about scaffold attachment regions and gene stability. Scaffold attachment regions are the regions of DNA isolated based on their ability to bind to the nuclear scaffold. Protein depleted chromosome forms a central scaffold surrounded by DNA. SARs are known to stabilize gene expression in transgenic plants. During the course of transformation, SARs are ligated to the flanking regions of foreign gene. It is observed that the transformation efficiency by particle bombardment is increased by use of SARs. The presence of SARs along with transgene stabilizes or normalizes gene expression. Example SARs from yeast and tobacco have helped to stabilize gene expression in plants. The degree of stabilization of genes by SARs is dependent on the affinity of SARs towards the nuclear matrix of the target plant cells. SARs are particularly useful for stabilizing the gene expression when the copy number of DNA introduced is high. In the absence of SARs, high DNA copy number would result in an unstable and highly variable gene expression. Now let's know about introns and gene expression. The presence of introns between the promoter and coding regions of a gene will significantly influence the gene expression. The gene expression is enhanced when introns are used in monocotyledonous plant species. For instance, introduction of introns between cauliflower mosaic virus 35 phase promoter and glucuronidase significantly improved gene expression in maize. It is believed that introns may stabilize mRNAs and increase the protein biosynthesis. There are conflicting reports on the effect of introns in the gene expression in dicotyledonous plants. Some workers have reported stimulations while others contradict these claim. It is now accepted that the intron containing constructs will enhance gene expression in transgenic monocots, although the picture is less clear as regards dicots. Gene silencing. There are, in fact, many instances of gene transfer that are not properly expressed. The instability of gene expression in transgenic plants is referred to as gene silencing. The mechanism of gene silencing is not well understood, although it is predominantly due to the phenomenon of homology dependent gene silencing. Two types of gene silencing are known in plant biotechnology: transcriptional gene silencing and post transcriptional gene silencing. The two differ at the level of gene expression where silencing occurs. Transcriptional gene silencing. When the transgenes share homology in their promoter regions, TGS occurs. It is due to altered methylation patterns and altered chromatin conformation. Gene silencing occurs by repression of transcription. Post-transcriptional gene silencing. Let's know about post-transcriptional gene silencing. Sometimes the expression of a homologous transgene may inhibit the expression of the transgene and endogenous gene. This primarily occurs by decreasing the stability of RNA and thus a reduced expression. This is often observed when strong promoters are used to drive the transgene expression, which may result in co-suppression or PTGS and thus a lower level of gene expression. For this reason, some workers prefer to use weak promoters rather than strong promoters. Now let's know now post-transcriptional gene silencing is due to the production of double-stranded RNA either in the nucleus or cytoplasm. The double-stranded RNA is found when antisense RNA is produced due to activity of RNA-dependent RNA polymerase. Now let's know about the strategy to avoid gene silencing. The control of gene silencing is not an easy job since it occurs in an unpredictable fashion. Some general recombinations are however, made to minimize the impact of gene silencing in transgenic plants. Reduction in the number of transgenes inserted, that is, reduced copy number, avoiding the use of promoters and transgenes with a high degree of homology, minimizing or avoiding the use of multiple copies of same promoter or terminator. Gene silencing is not observed in chloroplast transformation, hence it is preferred in recent years. Now let's know about the chloroplast transformation. Chloroplast or plastids and mitochondria are believed to have evolved from prokaryotes during the course of evolution. Both these organelles have their own genome, although it is much simpler when compared to nuclear genomes. Further, many of the proteins that function in chloroplast and mitochondria are encoded by nuclear genes and then transported to the organelles. Now, let's know about genome. Most of the higher plants have about 100 chloroplasts per leaf cell. Each chloroplast contains approximately 100 copies of chloroplast DNA genome. The chloroplast genome is a circular double-stranded DNA molecule located in the stroma. Majority of chloroplast genomes are in the size of 120 to 160 kilobase pair and contain about 120 to 140 genes. About 100 chloroplast genes are known to code for proteins. The protein synthesis in chloroplast resembles that of prokaryotes. Chloroplast engineering. Genetic engineering of chloroplast that leads to chloroplast transformation is an important and exciting field in modern biotechnology as it follows some advantages. Let's know about these advantages. First is chloroplasts are maternally inherited. Hence, there is no danger of gene transfer through pollen to related weeds. This is because pollen does not contain transgenes. Second point is multigene transfer can be conveniently carried out in chloroplast, which is rather difficult with nuclear genome. Third point is chloroplast genome is functionally comparable to prokaryotic genome. A single promoter can control the expression of group of genes, that means transgenes. It is therefore possible to introduce desirable multiple genes which can be expressed under the control of a single promoter. Fourth, high level of transgene expression is possible with chloroplast. There are about 100 chloroplasts per cell, each containing about 100 copies of genome. Thus, there is possibility of 10,000 copies of transgenes per cell. This is a tremendous number of transgenes carried by transformed chloroplast. There is a tremendous potential for a very high level of gene expression and a large scale production of active proteins. Chloroplast transformation is not associated with gene silencing, which is a major problem with nuclear genome transformation. Antibiotic resistant genes need not to be used as selectable markers. Even if used, they can be easily excised. Toxicity associated with foreign protein production in chloroplast is much less when compared to nuclear controlled foreign proteins. Let's know about the design of vectors for chloroplast transformation. First point is a construct for expression of a single gene that that is the vector for chloroplast transformation is based on the selectable marker gene AADA that provides resistance to antibiotic spectinomycin. The single foreign desirable gene is fused to regulatory sequence, which in turn is flanked on either side by chloroplast DNA, ctDNA. Second is a construct for expression of multiple genes. In this case, the selectable marker is the betaine aldehyde dehydrogenase gene. It is flanked by a promoter and the multiple genes are flanked by a terminator. At both ends, chloroplast DNA sequences are present. In between the transgenes, there are ribosome binding sites to ensure efficient transformation. Now let's know about the introduction of foreign genes into chloroplast genome. Most of the methods used for introducing the foreign genes into nuclear genome are not useful for chloroplast transformation. The most successful method for inserting foreign genes into chloroplast is particle gun bombardment. After the bombardment, homologous recombination occurs between the chloroplast DNA sequences on the vector and those of on the genome. This is a site-specific integration and thus avoids the frequent problem associated with random insertion of foreign genes into nuclear genome. The regenerated plant derived from the modified plastome are regarded as transplastomic plants. The future of chloroplast transformation, the technology of chloroplast transformation is in the developing stage. In fact, it is not become as routine as transformation of nuclear genomes of plants. Chloroplast engineering, however, holds a great promise in plant biotechnology, being an efficient, clean, and environmental friendly approach for the production of transgenic plants. Thank you. Finally, I have finished genetic engineering of plants and its methods. Any question regarding this, you can ask me in the comment box and I will definitely answer the question. Please subscribe my channel for more updates. Thank you.